Hello and welcome to my channel. In this lecture we'll start talking about muscles, and we will begin with the skeletal muscles. So let's begin. All you know that the types of muscles in the body are of three types. The skeletal muscles, the one we use to move the body with, and the cardiac muscle or the heart muscle, and the smooth muscles that form the holy organs, the arteries, the veins, two of which are involuntary muscles, like the cardiac muscles and the smooth muscles. They are supplied by autonomic nervous system, while the skeletal muscles are voluntary muscles and supplied by somatic nerves. And there are differences between the criteria of all of the three types of muscles to suit their functions. This is another picture of skeletal muscles, smooth muscles, and you can observe the difference between the size of nuclei, for example. And the cardiac muscles, you observe the branching of the muscle fibers and the connections between each at what's so-called the intercalated discs. We will come to that later in details. The skeletal muscles, composed of muscle cells. The muscle cells are much longer than the other types of muscle tissue, and known as muscle fibers. And each fiber is composed of myofibrils, that is actin and myosin, as discussed later. The muscle tissue of skeletal muscle is striated because of the positioning of the actin and myosin molecules, and each striation is called sarcomere. It's the functional unit of the muscle. As mentioned before, the skeletal muscles are voluntary muscles and supplied by the somatic nervous system, while the cardiac and smooth muscles are involuntary muscles and supplied by the autonomic nervous system. This is a picture to show you a cut section of the muscle. Observe that it's composed of fascicles of muscle fibers, and each fascicle is surrounded by a layer of connective tissue called the perimysium. There's an endomysium that covers each muscle fiber, perimysium that surrounds the fascicles, and epimysium that surrounds the whole muscle. And observe, because of the positioning of the actin and myosin, because they differ in size. The actin are thin filaments, the myosin are thick filaments. That's why they produce the functional unit, which is called the sarcomere. And this is a molecule of actin and surrounded by myosin, and how tropomyosin, I mean, how they interact with the myosin molecule that has special neck-like projection ending with a head that cocks, on the actin, and produce the muscle contraction by using ATP. The skeletal muscle contains multiple fascicle. The muscle fibers, the fascicles of muscle fibers, formed from the fusion of cells called the myoblasts during development. The development process of the muscle is called the myogenesis. The cell has multiple nuclei. That's why it's multinucleated, and the nuclei are called myonuclei. So they're formed by myoblasts during myogenesis are multinucleated and contain nuclei. The nuclei are located inside the cell membrane, right next to the membrane, and because of the activity of the muscle fibers, they contain also multiple mitochondria to meet their energy needs during contraction. This is a microscopic picture of how a skeletal muscle looks like. See the striations because of the alternation of the actin and myosin molecules, and you can see the peripheral multi-nuclei in each muscle fibers. There are flat nuclei present inside the cell membrane. Like we said, the muscle fibers is composed of myofibrils, that is molecules of actin and myosin. They are called the myofilaments, and thrown into repeated units called the sarcomeres, which are the functional unit for the muscle contraction. And the power that the muscle uses to contract is derived from oxidation of fats and carbohydrates. This is aerobic metabolism, but also there is anaerobic chemical reactions that are also used, and the muscle starts to consume its stored glycogen. And this is particular in fast twitch fibers. The chemical reactions produced adenosine triphosphate, ATP, molecules that are used to power the movement of the myosin head. You can see these sarcomeres, the mitochondria scattered along the muscle fibers, covered by the sarcolemma, and you can see the peripheral flat nucleus. And we will come to the description of this sarcoplasmic reticulum and the triad in later slides. These are the system for release of calcium and transmission of signals inside the muscle fibers. Now, we said that connective tissue is present in all muscles. It's thrown into three layers: the epimysium that surrounds the whole muscle, the perimysium that surrounds the fascicles, and the endomysium that surrounds each muscle fiber. So epimysium, perimysium in between, and endomysium that surrounds the muscle fibers. Now there are two types of receptors inside the muscle. The muscle can't be lifted away like that without any sensors to detect the degree of tension and degree of contraction of the muscle, to protect the muscle by means of body reflexes. These receptors are the muscle spindles and the Golgi tendon organs. The muscle spindles has no origin and insertion attachment. They are thrown inside the fibers of the muscle, parallel to the fibers of the muscle, and composed of what's so-called extrafusal fibers and intrafusal fibers. Intrafusal fibers are the fibers inside the spindle itself. Extrafusal fibers are the muscle fibers itself. You can see the structure here. There is also a difference between the nerve supply of the extrafusal and intrafusal fibers. The extrafusal fibers are supplied by alpha motor neurons, while the intrafusal fibers are supplied by gamma motor neurons, and we studied this in the structure of the spinal cord. Once by its position, because of its location, once the muscle is stretched, These fibers fires back signals to the spinal cord to detect the degree of stretch of the muscle, and if stretch exceeds the limits, a reflex contraction occurs. And this is the basics of the jerk examination of the patient. You know, the knee jerk, the elbow jerk, and the ankle jerk. There are other types of receptors. Before we leave that, this is the muscle spindle, and this is microscopic picture of the muscle spindle. These are the intrafusal fibers and the extrafusal fibers. And this is a picture of the other receptors, which is composed of free nerve endings of nerve fibers turning into a special type of receptors called the Golgi tendon organs. It's called Golgi tendon organs because it's present at the myotendinous junction and informs the body about the degree of the contraction or the tension developing inside the muscle. Once it fires back, it produces reflex relaxation of the muscle. So it opposes the action of the muscle spindle. The muscle spindle, once it fires, it produces a reflex contraction of the muscle. The Golgi tendon organs produce reflex relaxation of the muscle to protect the muscle from over contracting. Now, the muscle, to transmit its power to the bone it works on, it's attached to the bone at a site called the myotendinous junction, and it's the muscle tendon interface. Force is transmitted from the sarcomeres in the muscle cells to the tendon, and so to the bone it's attached to, and movement occurs. Arrangement of muscle fibers, they have different arrangement, but you can classify the arrangement into according to the direction of fibers in relation to the axis of force generated by the muscle, into parallel muscles or parallel fibers, and pinnate muscles or pinnate fibers. Pinnate means feather-like, means it has a stem that the muscle fibers are attached to. Now, in parallel muscles, the fascicles run parallel to the axis of the force generated. But the relationship between the fibers can vary. You can see here circular muscle fibers, parallel muscle fibers, straight muscle fibers, convergent muscle fibers. These are all parallel, the types of parallel muscle fibers. Variations in the direction of fibers can produce fusiform muscles, strap muscles, and convergent muscles. Convergent muscles, like this one for example, is fan-shaped, and the muscle fibers, they fan out from the origin and converge at its insertion, and are fanned out broadly from the origin. Now, there are other types of parallel fibers, but thrown into a circle fashion, like the orbicularis oculi and the orbicularis oris, in which the fibers are longitudinal arrangement and create a circle from the origin to the insertion. According to penation, we said the muscle fibers run at an angle to the axis of force of generation, and the types of pinnate muscles are unipinnate muscles, bipinnate muscles, and multipinnate muscles. Unipinnate muscles has angled fibers that are at one side of the tendon, like when you study it in the upper limb, for example, the flexor pollicis longus is a unipinnate muscle. Bipinnate muscle fibers on the two sides of the tendon, like the rectus femoris, for example, and the multipinnate muscles fibers that are oriented at multiple angles along the force generated. They have multiple tendons around which the fibers are arranged, like the deltoid muscle. And it's believed that, and the soleus muscle also is multipinnate muscle. These muscles are famous of their strength. Penation produce strength in the action of the muscle. That's enough for this lecture. I hope you got the maximum benefit from this lecture, and thank you very much for watching.